Right, thank you. I hope everybody is still awake. I'm going to talk about ocular melanin and its role and how it impacts the ocular pharmacokinetics and drug disposition of various therapeutic agents that we use. I personally feel that ocular melanin is a key component in ocular drug delivery. Most of the times I think it's less explored area, and it's a part of equation, but unfortunately it's not investigated as much as one I would at least like to explore. So number of people have contributed to this work. I cannot take credit for what I'm going to present just by myself. So thank you for all of them. It was an excellent team effort between pharmacology team, chemistry team, biology team, and analytical science team. Very briefly, melanin is present in various parts of the body, and I'm going to focus my talk on just ocular melanin. So it is present in front and back of the eye, and it's a heterogeneous biopolymer. Basically, it has pyrrole ring and free carboxyl group and phenolic hydroxyl groups in it, synthesized from L-dopamine, tyrosine, and cysteine in the enzymatic process that are involved, and then various polymerization steps. Melanin, basically in the eye, the function of melanin is that it acts as a free radical scavenger. So basically it protects eye from UV lights, and it exists mainly in two forms. It's eumelanin and pheomelanin. Basically depends on what is the starting moiety of this melanin synthesis. That's where the nomenclature comes from. Pheomelanin is normally considered as a red melanin, and eumelanin is black melanin. So the eye color is basically either red or black, and anything in between of those two shades, depending on how much pheo and eumelanin you have in your iris. So that would give you the color of the iris. It has been observed that in uveal tract, basically iris choroid tissue, you would have more pheomelanin, and that's why it's more reddish as compared to retinal pigment epithelium that has more eumelanin, and that would give it its darker color. Some in vitro studies suggested that more eumelanin you have, more antioxidative properties one can have in the eye. And there are some controversial reports suggesting that the lighter color eye person may have higher risk of developing uveal melanoma or AMD, and this could be just as a result of less protection from the UVI. But that statement is still controversial, and there is no clear agreement on that statement. And in the cartoon that I have shown here, that's the concentration of melanin in various tissues. So melanin is highly concentrated in iris, front of the eye, and that constitutes 17% of the total uveal tract. And in the back of the eye, you would have around 40 micrograms per milligram of tissue. So that's the concentration reported in various literature. So significant amount of melanin is present in the eye, and that warrants us to investigate what's the role and how it would impact the drug delivery. A little bit more about melanin. This was reported back in 1980s that melanin content with age, it goes down significantly, specifically in human retinal pigment epithelium cells. So this could be the reason that with age we do not have that much of UV protection or some kind of radiation protection, and we start seeing the effect, and that may result into various ophthalmic diseases. By the way, it doesn't matter what color of the eye, in all different types of eye colored people, melanin content in retinal pigment epithelium goes down with age. One more interesting fact on melanin is that macular region, it has lower melanin content as compared to peripheral region. So if we talk about AMD, which is mainly a disease for macula, or many of the vision threatening disease that impact macula, this is something very interesting, that it has low melanin level as compared to peripheral region of retinal pigment epithelium. And as we see with age, in peripheral region, you would the melanin content goes down, as you see in this graph, but in macular region, the content remains constant even though it is low. And one last preclinical fact, that melanin content varies a lot among different preclinical species, and even same species but between two different strains, it varies a lot. So this was study published by Compella's lab, and what he has shown is that in monkey, if you look at choroid and RPE, you would have significantly higher amount of melanin as compared to rabbit or human, which is strikingly different content. So if we are looking for ocular drug disposition of drug that has a high melanin affinity, you may see completely different pharmacokinetics or ocular distribution of that particular drug in preclinical species than you would see in humans. So one should be really careful about translating PK data, distribution data, or even efficacy data from preclinical species to humans. And this could also explain the disconnect between efficacy that you see in preclinical species, but then you don't see in humans. So with that background, as I have already covered title of this slide, that it really impacts drug disposition in the eye, particularly the PK, and we have in the literature you see number of drugs that have been reported, as well as even at Novartis we have seen this internally in-house, that once you have a higher melanin affinity binding drug, you would see completely different PK profiles. So at the end of the day, when you change drug disposition or pharmacokinetics, nobody really cares about actual pharmacokinetics. The key from the FDA perspective or agency perspective, as long as your drug is safe and effective, those are the two key criteria. But both of them are very strongly related with PK of that particular drug. So if you have high melanin binding drug, you may have very low free fraction of drug available for the action. So that could impact your efficacy. And at the same time, with longer retention of these drugs in the eye, that may cause, with multiple dose administration, that may cause some local safety concerns as well. So these are few things that we would like to keep in mind as we go through drug development and the process. Here is the cartoon. I hope it works. I just made a cartoon that when you have low affinity drug, that would come out really quickly, relatively speaking, from the medium affinity drug, and as compared to high affinity drug, that would really once it reaches the uveal tract after whatever out of administration, it would have significantly different half-life or AUC, or mean residence time in the eye, or the rate of appearance in plasma would be very different. So with that background on melanin, on this slide what I'm showing you is a case study where we had one compound that we were profiling for a wet AMD project. And what we saw with this drug is that when we test ocular PK of this drug in pigmented rats, so we used Brown-Norway rats and non-pigmented rat, which is Sprague-Dawley male rats, and we dosed this drug orally 10 mpk dose, we see completely different ocular PK profiles. As you see in posterior eye cup of rat after oral dosing, with this line shows the PK profile in pigmented rats, and here is the albino rats. So it's a log scale. So there's at least, I guess, 6 to 7-fold difference in the area under the curve for posterior eye cup. And if you look at the retinal concentrations in pigmented eye, you would have significantly high, almost over 50-fold higher AUC than your albino or non-pigmented rats. So this clearly shows that when you have drug that has high affinity for melanin, you may see completely different profiles. So this triggered an internal exploratory work where we started looking into some kind of high throughput technique that we can implement in our drug discovery process, that when we profile various compounds, normally we start with millions of compounds in the library, and we go with thousands of compounds for in vitro studies and hundreds of compounds, or 10 to hundreds of compounds for in vivo studies. So we cannot, when we start with thousands of compounds in the drug discovery process, we cannot treat them, we cannot run the study that I showed, in vivo studies. So we need to have some high throughput technique to quickly but reliably screen these compounds that we can move forward, or at least we can assess the risk of these compounds and we can eliminate from the screen. So with that thing in mind, we looked into the literature, that what kind of techniques are available that we can utilize to quickly screen these compounds. And in the plasma protein binding field, that has been widely reported that there is a technique which is HPLC affinity based technique. When you have HPLC column, your stationary phase is plasma human serum albumin, and you would pass your compound or elute your compound from the stationary phase. Depending on the retention, depending on the affinity of your compound, the drug would come out at various time points. So you would have a retention factor. A higher affinity drug would come out later. So that's how you would relatively assign an arbitrary unit to your compounds, and you would rank order them for their affinity. And what in the literature they have shown that plasma protein binding, and when you do traditional way human serum albumin binding, there is a pretty good correlation when you use the HPLC assay based method, which is very quick and high throughput technique. So learning from that, we thought we would also use a similar technique where we would have stationary phase with melanin, and we can utilize that method. Unfortunately, it is not commercially available. So we had to custom made our columns after number of we had to go through number of problem solving issues, but at the end we could make custom made melanin columns, and we tried various high binders and low binders that are reported, and we have also included medium binders, but unfortunately that study did not go well, and we had some issues. So I don't have that data right now, but what we did is that we essentially ran similar way our study, and we could get a relatively linear curve with 0.9 R square value, where we have on the x-axis you have retention time, and the y-axis is the log k that we assign based on the retention time. When you have different high binders to the melanin, that would have longer k value or a higher k value, meaning that they elute out much later as compared to low binding drug that has lower affinity and that would come out fast. With this initial success, we also wanted to make sure that the assay we have is also robust, and columns that we prepared we could only use for a certain number of times. So we wanted to make sure that the column to column variability would be not a factor. So we tried bunch of columns, we prepared them, and we ran number of compounds with 2 different columns. I have data here, column 3 and column 4, but we have tried I think 5 to 6 different columns, and we see good correlation between 2 different columns when we use for the same compounds. So we have good confidence that the columns that we have prepared and the way we are running the assay, it's a robust and reliable assay. We get similar numbers no matter what columns we use. And the data in the right here, we wanted to compare this high throughput technique with more traditional way of running the melanin binding assay. That is shown in blue here. Basically, traditional method is a simple equilibrium method that we do for plasma binding as well, but that is a time consuming process. It takes lots of material. In the discovery phase, you don't have that amount of material to run full scale concentration dependent equilibrium binding assays. And but we had data for number of compounds, and we used the newly developed assay and we rank ordered the compounds. So here is the data in blue with traditional method, and data rank order in the green with the newly developed assay. As you see, they do not align exactly with each other. However, if you look at the bigger trend, when you have higher affinity, they pretty much both methods they show that they have higher affinity, medium affinity and lower affinity. So they are not completely random and they are not aligning 100% with each other. But all we are trying to see at this point of drug discovery is just the trend, whether they have high affinity, medium affinity, or low affinity. So we are using a very broad bucket to just put the compounds and rank order them. So we felt pretty confident that we can, at this early stage, we can still use the high throughput technique, considering all the advantages that it has to offer. And then we went ahead and we have done almost over 300 compounds now, and we have used these methods for rank ordering the compounds for their melanin affinity. So the graph I am showing you here is that molecular scaffolds. We have used different compounds with different chemistry and different molecular scaffolds. So the molecular scaffolds, we have observed that it influences melanin binding affinity. As you see, the reds are with higher melanin affinity. They are from the similar type of molecular structures, and the blues are all together. The oranges, they are all in the medium affinity range, or greens are in lower affinity range. So molecular structures, and we have also done lots of in silico modeling, and we have also worked on coming up with some kind of molecular descriptors that are responsible for melanin binding. It's a subject of completely different and new presentation. There was a lot of work done to come up with molecular descriptors and properties of molecules that can define its affinity, or you can predict its binding affinity for the compounds. But I'm not going to talk about that at this point. So for my work, what we have done is that in order to validate the method, because this is still in vitro, we have selected 3 different compounds: high melanin, high affinity compound, medium affinity, and low affinity compound. And we have done in vivo studies and see if this in vitro assay would translate into in vivo studies. So basically, three compounds with different affinity, and either we tested them in albino rats, which has no pigments, or Brown-Norway black rats that has pigment, and then the PK studies. So there it is. What we have observed, I would like you to just focus on the left curve right now. What you have here on the x-axis is 3 compounds with its log K value that comes. These values come from the in vitro assay. So 0.54, that is low melanin affinity compound, 0.84 with medium affinity, and 1.27 with high affinity compound. And on y-axis, you see that is fold increase in the area under the curve of this compound between Brown-Norway rats, which is pigmented, over non-pigmented rats. So when you have low affinity compound, you do not see significant difference between two species. However, as the affinity goes up, you start seeing the difference in their PK and in their AUC values. Specifically for high affinity compound, if you see, you have almost 60-fold higher area under the curve for high affinity compounds. Drug staying longer in the eye, meaning choroid, and giving you higher AUC values. And the curve on right hand side is only for the high melanin binder that you see here. This is the PK profile. The plasma is not significantly different between two species, so the difference is not just because of the plasma exposure. However, if you look at the posterior eye cup concentration, significantly higher in pigmented animals as compared to non-pigmented animals. So this gave us good confidence that we see a trend that is coming from in vitro assays, and we could translate into in vivo studies. So this was very promising results for validating the in vitro assays. So in summary, what we have seen so far is that melanin binding can impact and it can modulate ocular pharmacokinetics. We have also validated in vitro method with in vitro studies. And in future, we also plan to run knockout mice so that we can have same background, because there is a question that if they have these are two different strains, so they may have some different metabolism or different anatomy, then that could be interfere with your interpretation. So by using the knockout mice, we may have same genetic background, and we would have only difference in their ocular pigments, and we can easily differentiate the effect. As far as the take-home message is concerned, or how do we use this data, so the key applications of these findings to the drug discovery or development is, first, I think it's very important to look at the drug melanin affinity early in the discovery space, and you can utilize in vitro assays. If you find that drug has high affinity for melanin, then you can check it for its reversibility and what would be the impact on ocular PK and plasma, where you can do in vivo studies. If the drug is really promising but has high melanin affinity, you may also want to run some whole body autoradiography studies and find out the distribution in various parts of the body, and especially in brain. And if you also want to share your knowledge in your preclinical safety groups and translation medicine colleagues, so that they can modify their protocol in their safety studies, and they can include at least one pigmented species to assess the risk of high affinity compounds. And as I mentioned earlier, the species and strain specific difference that we see in preclinical spaces, it's very hard to translate into human data. So one needs to be really careful about how to take this data and translate into the humans. With that, I have a number of people to acknowledge. All my pharmacology team, chemistry colleagues, analytical science chemist colleagues, and computer-aided drug discovery team. With that, thank you very much for your attention. Any questions?
Yeah, Dr. Mishra.
Yes, melanin binding could be reversible or irreversible. And when you have reversible, you are going to get drug into the aqueous humor or vitreous when the drug eliminates, reach equilibrium. But if it is irreversible, then you're going to drug concentration will go down pretty soon.
Yes.
So in both cases, the free drug is the one that is going to do, effect in the retina or iris or wherever it is. So how you're going to correlate the efficacy of the drug with the percentage bound with the melanin, either reversibly or irreversibly. Have you thought about how, like the plasma protein binding, the higher the binding, the less is the free concentration and the effectiveness goes down?
Yes. So, following the same concept, if we do it, if we have a number, first of all, if it is reversible, it's less of a concern. If it is irreversible, then obviously efficacy would go down. I think the key application of that number would be when we use modeling and simulation data. You can predict that can be one parameter that you would want to include in your modeling and simulation as you predict your efficacy. So
even with the reversible binding, if 99% is bound, you have only 1% remaining all the time.
Yes.
As it's depleted, it will come back, but you are not going to get free drug concentration more than 1%.
Yes.
So that is going to also, your target concentration may not be reached for whether you're doing AMD or diabetic retinopathy. It's going to be difficult.
It's possible, and that is why we would like to screen this as early as possible. If it is really high affinity compound, we would deprioritize the compound. I think medium affinity would be the one that I would take it forward. Medium to high, but not too high affinity compound. So this would
second quick.
It's just a comment coming from a pigmented race. It's a very good study because it fills in a lacuna which was not there before. Most of the drug formulations have been titrated for less pigmented eyes. So especially as a glaucoma person, I would be interested to know where the trabecular meshwork is more pigmented in our race. Would this have further implications vis-a-vis the free and bound to melanin? If it's more bound to melanin and in trabecular meshwork is more pigmented, then you can target the tissues of at least for glaucoma like that.
Sure, it can be used at our advantage as well.
Yeah.
You can make a depot and then have the drug released over a longer period of time. So it all depends on the kinetics of the binding. What is the on rate and off rate of your compound to the melanin?